we'll speak about main types of the chemical conversions and extremely important coenzymes. We already know some types of these chemical conversions. First one is transamination. It means changing in the functional groups. It is transfer of amino group of one amino acid. It's conditional picture. Transfer of amino group from one amino acid to keto acid carbon skeleton. It is reversible conversion. There are two substrates in the transamination. One is amino acid, second one is keto acid. Sorry, let me correct it. It's oxygen, it is keto group, it means keto group. And there are two products in the reaction of transamination. It is new keto acid, and the second one is new amino acid. Common acceptor of amino groups for the most amino acid is known compound, which is alpha ketoglutarate. It is metabolite of the Krebs cycle. A second reaction here is deamination. It is direct elimination of the amino group with the formation of the ammonia ion. There are several types of deamination. It can be intramolecular deamination, like for histidine, or hydrolytical deamination, like for serine and threonine, and oxidative deamination of glutamic acid, of course. Decarboxylation means, let's add this thing, elimination of ammonia, amino group in the form of ammonia. Decarboxylation means elimination of carboxyl group in the form of CO2. It leads to formation of biologically active compounds, which are called biogenic amines. Hydroxylation is incorporation of the OH group into the structure of the molecule. And one carbon unit transfer leads to formation of new compound. Example is if we take 3 carbon compound or 2 carbon compound glycine, we can produce 3 carbon compound serine as a result of this type of conversion. Now we'll speak about 5 reactions in details and 3 main coenzymes. There are 3 main coenzymes in the metabolism of amino acid. First one is PLP. Do you remember full name of PLP? Could you answer?
Phosphorate lip.
No. Precursor.
Pyridoxal.
Yes.
Yeah, continue, please. Pyridoxal.
Pyridoxal.
Phosphate. Pyridoxal phosphate.
Pyridoxal phosphate.
Phosphate.
Precursor of pyridoxal phosphate is water-soluble vitamin B6, pyridoxine. Yeah, phosphorylation of pyridoxine leads to formation of pyridoxal phosphate. Second coenzyme here is called tetrahydrobiopterin. Tetrahydrobiopterin is a new cofactor for us. Sometimes this type of the abbreviation is used in another alternative textbooks, BH4. This abbreviation can be used. Tetrahydrobiopterin participates in the reactions of hydroxylation of the ring in the structure of metabolites intermediates. And the last one here is called tetrahydrofolic acid. This abbreviation in brackets, tetrahydrofolic acid. It is coenzyme.
It is B9.
of folic acid. Yeah, folate is precursor of
Yes.
tetrahydrofolic acid. Now let's discuss the types of conversion and participation of these cofactors, coenzymes in this reaction. First one on this slide is transamination. Transamination, first type of reaction, and coenzyme PLP, which plays the role of temporary acceptor of amino group. At the left, you can see two substrates. First substrate is amino acid. 17 amino acids can undergo transamination, except 3 of 20 proteinogenic, lysine, proline, and threonine. Other 17 can participate in the transamination. Again, one substrate is amino acid with amino group in the structure. Second substrate is keto acid. Here you can see alpha ketoglutarate. For the most reactions, it will be alpha ketoglutarate. At the right, you can see two products. First one is keto acid, new keto acid, yeah, and new amino acid, which is glutamate. We can say it is transfer of amino group to keto acid forming new amino acid, but really it is more complex. Firstly, amino group is transferred to pyridoxal phosphate to form intermediate pyridoxamine phosphate, and later this amino group is transferred to carbon skeleton of keto acid to form new amino acid. Enzyme, common name of enzymes is aminotransferase or transaminases. Real name depends on the substrate. If L-amino acid is aspartic acid, enzyme is called aspartate aminotransferase. In the case of alanine, it is alanine aminotransferase. Example. On the next slide, I want to show you precursor of pyridoxal phosphate, vitamin B6, the structure of B6. It has the ring in the structure. It is heterocyclic compound, nitrogen-containing ring. Phosphorylation of pyridoxine. Phosphorylation leads to formation of our PLP, pyridoxal phosphate. Pyridoxal phosphate. Al in the name means an aldehyde group. Aldehyde group of this coenzyme participates in the formation of the linkage with the enzyme with aminotransferases. PLP is a coenzyme of deamination. On this slide, you can see the scheme of alanine and serine conversion to pyruvate. Both amino acids, alanine and serine, both contain 3 carbon atoms. The carbon skeleton consists of 3 carbons only. Both are converted to pyruvate. Alanine undergoes transamination. Reaction number 1 is catalyzed by alanine aminotransferase. And new keto acid, which is formed in this transamination, is pyruvic acid 3 carbon compound. Serine, reaction number 2 on this slide. Serine undergoes deamination. It means elimination of amino group in the form of ammonia. It's better to add NH4 plus. Direct elimination of ammonia, and it is hydrolytical deamination. Yeah, OH group and H group are eliminated in the form of water. Serine dehydratase catalyzes this reaction, and serine dehydratase and alanine aminotransferase both require participation of PLP as a coenzyme. They are PLP-dependent enzymes. On this slide, you can see utilization of carbon skeletons of alanine and serine in the form of pyruvate. We already know that pyruvate later is converted to acetyl-CoA to be used in the Krebs cycle for the synthesis of energy. Here I want to show you another dehydratase. It is deamination again. It is hydrolytical deamination of threonine. Threonine is OH containing amino acid. OH containing amino acid. Threonine dehydratase catalyzes elimination of amino group in the form of ammonia and water, and threonine dehydratase is PLP-dependent enzyme. Threonine has 4 carbons in the linear chain in the structure. Therefore, alpha-ketabutyrate is formed, new keto acid, alpha-ketabutyrate. Alpha-ketabutyrate is converted to propionyl-CoA as a result of decarboxylation. Carboxyl group is eliminated in the form of CO2. Alpha-keto acid dehydrogenase catalyzes this conversion. I hope you remember conversion of propionyl-CoA to succinyl-CoA. This part of the threonine utilization was discussed in the previous topic, lipid metabolism. Propionyl-CoA is a result of the beta oxidation of odd-numbered fatty acids, 3 carbons compound. 3-carbon compound is converted to 4-carbon compound methylmalonyl-CoA with participation of 2 very important enzymes, carboxylase and mutase. Carboxylase, let me open other slide to show you this way in details. Yeah, propionyl-CoA carboxylated using CO2. Enzyme name is propionyl-CoA carboxylase. It is biotin dependent enzyme. And this conversion is ATP dependent. ATP is used to incorporate 1 carbon into the structure of propionyl-CoA to form methylmalonyl-CoA, D form of methylmalonyl-CoA. This compound requires isomerization, epimerase firstly, to form L-methylmalonyl, and finally mutase, which require participation of B12, cyanocobalamin, to produce succinyl-CoA. The fate of succinyl-CoA is TCA, tricarboxylic acid cycle, this intermediate of the Krebs cycle. Do you have the questions about transamination and deamination? Let's summarize. Transamination is transfer of amino group of amino acid to keto acid to form new keto acid and glutamate. We can say new amino acid, but primarily it is glutamic acid because alpha-ketoglutarate is used more often than other keto acids in the reactions of transamination. Second type is deamination, elimination of amino group in the form of ammonia. We discussed hydrolytical deamination of serine and threonine in detail. See, threonine 4-carbon compound, conversion of to alpha-ketabutyrate, 4-carbon compound, decarboxylation, and carboxylation again to get finally succinyl-CoA. This is way for utilization of threonine. I told you that in this colloquium we'll speak about synthesis of amino acids and the utilization of amino acids. In the case of threonine, threonine is essential amino acid. It means threonine cannot be synthesized in our cell, but we have to know all about utilization of threonine, utilization of carbon skeleton of threonine. This way means conversion of threonine to succinyl-CoA to be oxidized in the Krebs cycle. If no questions, let's continue. Next type of the chemical conversion is called decarboxylation. In accordance with the name of this type, we have to understand it is elimination of carboxyl group in the form of CO2. On this slide, you can see decarboxylation of two amino acids. One is glutamic acid, glutamate. Second one is histidine and glutamate. Decarboxylation of glutamic acid, it is decarboxylation of alpha carboxyl group. I want to remind you, glutamic acid has two carboxyl groups. One is alpha because of it is linked with alpha carbon. Another one is gamma carboxyl group. Alpha carboxyl group of glutamic acid is eliminated in the form of CO2, forming GABA. GABA is an abbreviation. Full name of this compound is gamma amino butyric acid. Now, alpha, beta, gamma carbon amino group is linked with gamma carbon, therefore gamma amino butyric acid, or GABA in abbreviation. This reaction is catalyzed by glutamate decarboxylase. It is PLP-dependent enzyme. Again, PLP in transamination, PLP in deamination of the most amino acids. And here you can see participation of PLP in the decarboxylation of amino acids. GABA is one of the brain's major inhibitory neurotransmitters, being released at 30% of its synapses. A GABA inhibitory neurotransmitters. Second reaction on this slide, it is decarboxylation again. Initial compound is histidine. Histidine is decarboxylated under the action of histidine decarboxylase. It is PLP-dependent enzyme. A carboxyl group is eliminated in the form of CO2, forming next another amine. It is called histamine. Histamine is involved in allergic responses and in the control of H-chlor secretion by the stomach. It is a regulatory molecule. Both GABA and histamine can be called biogenic amines. They are amines with biological activities. One of them is neurotransmitter. Histamine participates in the formation of allergy reactions. Antihistamine drugs prevents formation of histamine acting on enzyme. Therefore, they prevent formation of allergy reactions. Let's continue with next coenzyme. Next coenzyme is called tetrahydrobiopterin. This coenzyme is required for ring hydroxylation. Hydroxylation of the rings require participation of molecular oxygen, O2. One oxygen atom is used for formation of OH group in the product. The second one is found in water. Let's discuss one example. At the right, you can see the structure of phenylalanine. Phenylalanine is proteinogenic amino acid. It is essential amino acid. It cannot be synthesized in our cells. Hydroxylation of phenylalanine leads to formation of non-essential amino acid tyrosine. The difference between phenylalanine and tyrosine is OH group in the structure, which is incorporated into the aromatic ring of phenylalanine. Precursor of this OH group is molecule oxygen. Here you can see this oxygen. One atom of oxygen is used for the synthesis of this OH group. Second one, firstly, is transferred to pteridine ring of tetrahydrobiopterin. Here you can see the structure of tetrahydrobiopterin and positions nitrogen and oxygen, the ring which participates in the formation of water. Firstly, second oxygen is transferred to pteridine ring. After that, by dehydratase, water is eliminated using OH group of pteridine 4A carbonylamine, and this hydrogen, neighbor hydrogen, forming dehydrobiopterin. Dehydrobiopterin undergoes reduction by the action of reductase using NAD or NADP, and tetrahydrobiopterin can be formed. It is not necessary to memorize the structure of tetrahydrobiopterin. We will use the abbreviation. Yeah, we have to know that conversion of phenylalanine to tyrosine require participation of tetrahydrobiopterin, and we will mark BH4, tetrahydrobiopterin, BH4, is converted to BH2. Yeah, we will add water. We will make our scheme easier for us. Yeah, we have to know that one oxygen from molecular oxygen is incorporated into the structure of tyrosine. Another oxygen is used for formation of water. Donor of two hydrogens for water formation is tetrahydrobiopterin, which is converted to dehydrobiopterin. It is possible to regenerate dehydrobiopterin to tetrahydrobiopterin using reductase and donor of two hydrogens, which can be NADH H plus or NADPH H plus. Here you can see the ring of tetrahydrobiopterin, which is called pteridine ring. This ring similar to part of isoalloxazinic ring. Isoalloxazinic ring is component of B2 riboflavin. This part similar to pteridine ring of BH4 ring. Here for nitrogen, two fused rings for nitrogen like in B2. Both vitamins, coenzymes, it's better to say coenzymes, both coenzymes, FAD and BH4, participate in the oxidation reduction reactions. Can be used as donors and acceptors of protons and electrons. Questions, please?
Everything is clear.
Okay, thank you.
Clear,
Let's,
ma'am.
yeah, okay, yeah, questions, yeah? Oh, clear, yeah, okay, thank you. Let's continue. Um, let's discuss conversion of tyrosine. In previous slide, in the course of the information in previous slide, we already know that phenylalanine is converted, or it is precursor of tyrosine in our cells. We have to know that tyrosine is precursor of epinephrine, dopamine, non-epinephrine, and it is used for formation of T3 and T4, a lot of functions. How it is possible to convert tyrosine to epinephrine? Here you can see the set of the chemical conversions which lead to formation of epinephrine in adrenal glands and adrenal gland medulla. Tyrosine is converted to DOPA. DOPA is abbreviation, full name is dihydroxyphenylalanine. Really, it is phenylalanine with 2 hydroxyl groups. Second hydroxyl group is incorporated into the structure of tyrosine in the same manner like in previous slide. Again, precursor of this green OH group in the structure of DOPA is molecular oxygen. Tetrahydrobiopterin is used as a donor of 2 hydrogens, and byproduct in this reaction is water. This reaction is catalyzed by tyrosine hydroxylase. I forgot about enzyme in previous slide. Let's go back. Yeah, phenylalanine hydroxylase. And here now tyrosine hydroxylase, same manner of hydroxylation using molecular oxygen and tetrahydrobiopterin. In the next step, carboxyl group of DOPA is eliminated in the form of CO2 under the action of aromatic amino acid decarboxylase, forming dopamine. Dopamine is used as a neurotransmitter. Next step, dopamine is oxidized again using molecular oxygen, but OH group now, the red OH group, is incorporated into the linear part of the molecule, and the donor of 2 hydrogens is ascorbic acid, reduced form of ascorbate, which is converted to dihydroascorbic acid. Reaction is catalyzed by hydroxylase again, another hydroxylase which is called dopamine beta hydroxylase because of position of the OH group. Non-epinephrine is formed. Non-epinephrine is direct precursor of epinephrine. The difference between 2 molecules is methyl group, blue methyl group here. This reaction is catalyzed by methyltransferase N because of linkage between nitrogen and methyl group and methyltransferase. And the question is, what compound is used as a donor of the methyl group? This compound is called S-adenosyl methionine. We discussed S-adenosyl methionine in the synthesis of complex lipids, phosphatidylcholine, using phosphatidylethanolamine as a precursor. It is methylation. And here you can see methylation of non-epinephrine, which leads to formation of the epinephrine. Do you have the questions? Should we continue?
Just to mention what the compound donor of methyl group, which one?
S-adenosyl methionine, this one. S-adenosyl methionine. And in the next part, we will speak about synthesis of this compound from methionine to show you how it can be used and how it can be regenerated.
Thank you.
Other questions, please?
Yes, ma'am. Do we need to memorize those structures, like all of them, yes?
Yes, yes. We have to know phenylalanine, tyrosine, and all other intermediates till epinephrine. all of them should be found in your
Okay.
final test in colloquium. I think it is not so difficult if we compare the structure step by step. Yeah, if we know that it is the
No,
hydroxylation, decarboxylation, it seems to me it is not so difficult. Now let's continue. Let's discuss S-adenosyl methionine. What's this? Do you remember methionine? What's this? Could you explain me, please? What do you know about methionine?
Amino acid.
It contains methyl group, and
it is proteinogenic
it is
amino
essential amino
acid.
acid.
Of course, it is essential proteinogenic amino acid. It cannot be synthesized in our body, in our cells. It must be obtained with diet. It has sulfur in the structure. The functional group of methionine is called thiomethyl group. Thiomethyl group. Let's continue now with methionine. Want to remind you the structure. Here you can see methionine. Carbon skeleton of methionine contains 4 carbons, the main carbon chain. And this group is called thiomethyl group. Thiomethyl group. First step in the synthesis of S-adenosyl methionine is activation. Methionine interacts with ATP molecule, forming the linkage between sulfur of the methionine and carbon atom of adenosine part. 3 phosphates groups, all phosphates of ATP are eliminated. 2 together in the form of pyrophosphate molecule, 1 in the form of inorganic phosphate. S-adenosyl methionine is formed. We are speaking about this molecule. We can say it is activated methionine, or we can say it is S-adenosyl methionine, and we can use an abbreviation SAM, Mr. Sam, S-adenosyl methionine. The pink methyl group of this construction, pink methyl group, is used as a donor of the methylate in the reaction of the methylation. It can be previous one, yeah, this one. Methylation of non-epinephrine to get epinephrine. It can be used in another methylation reactions, including synthesis of phosphatidyl choline from phosphatidyl ethanol amine. Then Mr. Sam lost methyl group. It is converted to S-adenosyl homocysteine. S-adenosyl homocysteine. In the next step, hydrolase eliminates adenosine part from S-adenosyl methionine, forming this molecule. This molecule is called homocysteine. Please don't confuse the structures of cysteine and homocysteine. Cysteine has 3 carbons in the structure. Homocysteine has 4 carbons. Both cysteine and homocysteine have SH group. A difference is quantity of carbons. Please don't confuse, don't mix 2 structures. Reaction number 4 on this slide is regeneration of homocysteine to get methionine again. It is possible using methionine synthase and another coenzyme, a cofactor, which is called tetrahydrofolate. Here you can see methyl tetrahydrofolate. This compound is used as a donor of one carbon in the conversion of homocysteine to methionine. This enzyme requires participation of two cofactors, two coenzymes. Second one is B12. B12, want to remind you, it is cyanocobalamin. Firstly, methyl group of methyl tetrahydrofolate is transferred to B12 to get methyl cobalamin, and methylcobalamin is direct donor of methyl group to homocysteine to get methionine again. This is the cycle of methionine usage and regeneration. Again, let's refresh it. Let's finish it. Summarize it. S-adenosyl methionine, Mr. Sam, S-adenosyl methionine, we can say it is activated form of methionine which can be used in the reactions of methylation. S-adenosyl methionine is formed as a result of the interaction between methionine and ATP, forming SAM. After the methylation, S-adenosyl homocysteine is formed. Hydrolytical cleavage of the bond between sulfur and adenosine leads to formation of homocysteine, and homocysteine can be converted to methionine using two cofactors. First one is methyl tetrahydrofolate, and second one is B12 cyanocobalamin. Both are temporary acceptors of the methyl group. Both participate in the transfer of one carbon to homocysteine to get methionine. Questions, please? Clear, okay. And the last set, the small set about tetrahydrofolic acid, about this cofactor. Don't worry, it is not necessary to memorize the structure of tetrahydrofolic acid. Not necessary to know the structure of S-adenosyl methionine. You have to know only full name and abbreviation and the functions. But please memorize homocysteine. On this slide, you have to know the structure of methionine and homocysteine. Only two structures. Now let's continue about tetrahydrofolic acid. Precursor of tetrahydrofolic acid is folic acid. We discussed folic acid in the first semester. It is water-soluble vitamin. It has very complex structure, and here you can see the structure. It has pteridine ring. Para means para-aminobenzoic acid. This fragment is called para-aminobenzoic acid. It is residue of carboxyl group. And glutamate is a part of folate, can be from one or for several residues of glutamic acid. This folic acid. Firstly, reductase, enzyme reductase, transfers hydrogens using NADPH, reduced form of NADP, to get tetrahydrofolic acid, tetrahydrofolate. This tetrahydrofolate is used as an acceptor and donor of one carbon fragments. Now we have to understand that tetrahydrofolate can be acceptor of one carbon unit in different sterols. Want to show you. In the next slide, you will see only fragment of tetrahydrofolate. Fragment, this one, this part. N5 and N10. N5, nitrogen number 5, is a part of the ring. Nitrogen number 10 is a part of para-aminobenzoic acid here, linear part of this structure. It is interesting because both nitrogens N5 and 10 participate in the formation of the linkages with one carbon fragment. Now, here you can see this fragment N5 and N10 of tetrahydrofolic acid. First form of the tetrahydrofolic acid is called N10 farmyl tetrahydrofolate. N10 because farmyl group, one carbon farmyl group, is linked with N10 of tetrahydrofolic acid. Elimination of water from this compound leads to formation of the bridge between N5 and N10. Therefore, this form of the folic acid is called N5 and 10 methynyl tetrahydrofolate. Methynyl, the double bond here. Reduction of this double bond leads to formation of another form. It is called N5 and 10 methylene tetrahydrofolate. Single bonds. In both cases, the bridge between N5 and N10 and so methylene tetrahydrofolate. And finally, reduction of this bridge leads to formation of N5 methyl tetrahydrofolate with methyl group, which is linked with nitrogen number 5. You have to understand what does it mean, N5 and 10, and the names of intermediates, methyl, methylene, methynyl, and farmyl tetrahydrofolic acid. Let's go back. I want to show you this reaction again. It is regeneration of our methionine after the methylation. Homocysteine interacts with the methyl group, donor of the methyl group, N5 methyl tetrahydrofolic acid. Methyl group is linked with N5 or tetrahydrofolate, one of the forms of tetrahydrofolic acid as a coenzyme, one of the forms. I'll send you this table. In this table, you can see different forms of tetrahydrofolic acid of one carbon unit. It is farmyl tetrahydrofolic acid, N5 and 10 methylene tetrahydrofolic acid, methyl, farmimino, and methynyl, all of them. You have to understand what does it mean each name and the role of tetrahydrofolate as a donor of one carbon. We discussed this role in the regeneration of methionine from the homocysteine, and finally I'll show you synthesis of glycine using serine with participation of methylene tetrahydrofolate. Serine is 3-carbon compound. It is non-essential amino acid, proteinogenic amino acid. It is used, this one is used for the synthesis of the glycine, which is 2-carbon compound. Let's mark it. 3-carbon compound is converted to 2-carbon compound. The question is, where is this carbon? The fate of this carbon. Tetrahydrofolic acid is used as acceptor of one carbon in the conversion of serine to glycine. This reaction is catalyzed by serine hydroxymethyltransferase. Really, it is reversible reaction, and glycine can be converted to serine again. Tetrahydrofolate can be used as a donor of one carbon fragment. If we are speaking about direction serine-glycine, one carbon of the serine is transferred to tetrahydrofolic acid, forming this form of tetrahydrofolate. It is called methylene tetrahydrofolate, the bridge between N5 and N10. This compound, methylene tetrahydrofolate, can be used as a donor of one carbon fragment in different reactions, including synthesis of purine nucleotides. Second reaction on this slide is cleavage of glycine. It is catalyzed by glycine, mitochondrial glycine cleavage system, it leads to formation of easiest simple